Thank you, Jean, for your generous introduction. It's my great pleasure to be here giving yet another Darcy lecture, it's number 61. And this particular talk is given 26 times. Darcy lectures are sponsored by the National Groundwater Research and Educational Foundation of the National Groundwater Association. And the mission of the foundation is to advance activities related to broader understanding of groundwater and groundwater problems. And I would like to invite you to visit the National Groundwater Association website to learn about many activities that the association has. I would like to acknowledge my co-authors and collaborators. This is a work that has been going on for more than 20 years, and I've been fortunate to work with former postdoc, former Ph.D. student, current postdoc, former Ph.D. student, former postdoc, and colleagues from different universities. What I would like to talk about is theories of two-phase flow. And we know that the cornerstone of theories of two-phase flow is Darcys Law. Darcys Law was originally 150 years ago to model the flow of water under 1D, the steady-state flow of pure incompressible water in saturated, homogeneous, isotropic, sandy soil under isothermal conditions. And the equation is, there is the equation given as a linear relationship between flow rate and the rate of, and the hydraulic gradient, where the hydraulic potential is defined as pressure head plus gravity, plus elevation head. And over the years, this has been said to be extended to apply to 3D, unsteady state flow of two or more compressible fluids with any amount of dissolved matter in heterogeneous, anisotropic, deformable porous media under non-isothermal conditions. And you would think that the equation is a lot more complex than the simple equation, and it's not. It's the same equation. The only thing that's changed are these superscript and subscript added there, because the definition of H is also exactly the same as the definition of H for a single phase. How is such an extension done? Well, this is a little bit provocative, but this is how my story would go. The original equation by Darcy was written actually in a difference form, and then if you are interested in a one-dimensional flow, you write it in a differential form. But this is a steady state. If it is unsteady state, all we need to do is say H is not just a function of X, but also time. So we have to replace that derivative with a partial derivative. And if it is three-dimensional, we write the equation in three directions, and all we need to do is add this index I. And if it is anisotropic, all we need to do is replace the scalar K conductivity by a tensorial conductivity, and the same equation. And if it is heterogeneous, again, all we need to say is that Kij is a function of space. Otherwise, we use the same equation. If it is unsaturated flow, we use the same equation, but now we say that the conductivity is a function of water content, a volumetric measure. If it is two-phase flow, then we write the equation twice, once for the water or the wetting phase, and once for the oil or the non-wetting phase. Three-phase flow, we write it 3 times, 4 times, and so on. And the only difference is that now we have these conductivities for each phase. These are smaller than the saturated conductivity, so the relative permeability is introduced, and it is said that this relative permeability is a function of saturation. So this relative permeability, and for each phase a separate relative permeability. And it is said the conductivity for each phase is now smaller than the saturated conductivity, so rel perm must be less than 1. I'll go back to this in a minute. So to me, there is no extension. we have added what I call bells and whistles to a simple equation to make it look more complex and therefore applicable to a more complex system. I would say one has to go the other way around. You have to have a complex theory applicable to a complex system, and then you have to make assumptions when you are dealing with a simpler system and reduce it to a simpler formula. And you would say, yeah, but what does it matter? That's philosophy. It works, then we should be happy. Well, now, first of all, it doesn't work. I'll talk about that in a minute. And secondly, philosophy in science is important. And I would like to illustrate that concept by an anecdote from a book called Physics for Poets by French. Beautiful book. You should read it even if you're not a poet. You will learn a lot about physics. And it's talking about the theory of free fall of objects. What has been the theory? Now, the original theory was by by Aristotle. Aristotle said that when you let an object fall, it will immediately reach a terminal velocity which is constant. Now, that is the theory of Aristotle. And this was there for many, many centuries until Galileo and some others before him. They came and said, no, that's not true. You let an object fall, the velocity is 0. And it will increase, and in fact, under, vacuum, it will increase linearly with time. So this is the theory of Galileo. And what do we find? Well, we find Aristotle's theory is much better. It works maybe in the very beginning, few millisecond doesn't work. Otherwise, it works perfectly. Well, if we had accepted that kind of mentality and philosophy, we didn't have theory of gravity by now. And then this is what French also says in his book. Then a typical Ph.D. research in physics would be for the Ph.D. students to go on top of tall buildings, let objects of different shapes and different density fall, and determine the terminal velocity as a function of mass density and shape of it. That's a Ph.D. in physics then. And you would think we are not making this sort of research. We've been doing it for decades and still doing it. How many capillary pressure saturation curves have we measured and published? And real perm curves, and residual saturation as a function of this, as a function of that. What is behind it? That is important. And that's why I think philosophy is important. But the equations, do they work? Let's see. These are the equations for two-phase flow, and you can actually write it for any number of phases, as I said. But let's stick to two-phase flow. So this equation are written twice for a wetting phase and for a non-wetting phase. And this is the extended Darcys Law, written in a pressure form. And this equation then substitute there gives you two equations, but you have three unknowns. One of the saturations, because the sum is 1, and two pressures. So obviously, we are short of one equation. This was, of course, recognized 100 years ago when two-phase flow equations were introduced. So they said, well, these three unknowns are related to each other. Pressure differences will be a function of saturation. And this was, in analogy with the microscale, was said that this is capillary pressure. And that's what we call it. But actually, this equation has never, ever been derived, and it cannot be derived, not for when there is flow. And secondly, it's really immaterial whether it's capillary pressure or not. The thing is that it is measured as Pn minus Pw, and that's how we use it. Okay, so this is suggesting anyway that there is an algebraic relationship between Pn minus Pw and saturation. And then there is also real perm. This is also said to be a function of saturation. Now, you would say this looks perfect. We have enough equation, and then there is an algebraic relationship. We can measure it. Then we are in business. Well, the problem starts when you look at these two equations. Let's take the first one. How is the capillary pressure saturation curve measured? You go quickly through that. I hope many of you know that. We take a soil sample, put it in a pressure plate apparatus of something similar. The sides are closed. We have two reservoirs in contact with this soil sample. And so this is, say, the non-wetting phase reservoir. Could be oil or air or any other non-aqueous phase. And here is water, or the wetting phase. Initially, if these two pressures are equal, and if this is filled with the wetting phase, with water, nothing will happen. So we can plot Pn minus Pw as a function of saturation. Saturation 1, pressure is equal, so the difference is 0, nothing happens. You increase this pressure a little bit, still nothing happens. And only when you surpass a threshold value, the entry pressure, then some water goes out and some non-wetting phase goes in. We wait until there is no movement of phases at all, and then we plot that pressure difference against this new saturation. That gives you another point. And then we increase that pressure again a little bit. Again, we wait until nothing goes out anymore. New saturation is obtained against that pressure difference. And we plot this, and we get a drawing curve or a drainage curve. Now, if this was the case, we had only one curve, would have been perfect. But if you now try to reverse the process, put water back in by increasing the water pressure or reducing the non-wetting phase pressure, then you don't get the same curve, you get a different curve. The data points fall on a different curve, very significantly different. And this is called a wetting curve. And if you are going to do this reversal halfway from drainage to imbibition or wetting and the other way around, we get yet other curves. They are called the scanning curves in drainage or in imbibition or wetting. So there is not just one single curve. And moreover, we are really measuring not the actual pressures inside, we are measuring the pressure outside the soil sample. And to get any of these curves, it takes a week sometimes for a soil sample. It takes a long time anyway. So you have an algebraic relationship only if we wait long enough. But then we use these curves for processes that sometimes takes an hour, much faster than what the time was needed to measure these curves. So something doesn't look right here. So you could ask the question, what happens if you measure these pressures? First of all, you measure them inside the soil sample, and secondly, you measure them as the process occurs and plot them as the process occurs, not wait until there is no flow. What do we get then? So we have done that kind of experiments. This is the soil sample. This is the same sort of apparatus. The soil sample is here. These are the two reservoirs. Here we use PCE, a non-aqueous phase. What was here extra? We had two pressure transducers here. One of them was hydrophobic, the other hydrophilic. So one of them measures the oil pressure or the non-wetting phase pressure. The other measures the water pressure. And you can measure the pressures as the flow occurs and then plot that. So we can do a static experiment, just like I explained now, or you can do dynamic experiment, which means that you increase this pressure to a large value and the process occurs within half an hour, and then plot the pressure differences. See what we get. So we have done that. These are the static curves. Right? We have primary drainage and the main drainage and main imbibition. And then we have done measurements where, as I said, we increase the pressure of the non-wetting phase to a large value. Here there are three different pressures, and we get different curves. These are data points fall along this curve. This is for main drainage. We can have main, that was primary drainage. We can have main drainage. We can have main imbibition. And if we plot everything together, we get this. So the blue, again, are static curves. It's, for example, the static primary drainage. But the dynamic primary drainage is totally different. And many people have obtained this also in soil physics. And the same holds for static main drainage, dynamic main drainage, and main imbibition in a static and dynamic. Very different. So what we're supposed to have? We're supposed to have a curve, an algebraic relationship. But first of all, we have this primary drainage curve. We have a primary imbibition curve, we have main imbibition, we have main drainage, we have main scanning curves in drainage, we have main scanning curve in imbibition, we can have secondary scanning curves in drainage and in imbibition, then we can have the dynamic curves in drainage, dynamic curves in imbibition. It's a mess. It's not one curve. So is it, you should really have a theory more than just saying Pn minus Pw is a function of saturation to be able to explain all of this. And what is that theory? How about the rel perm curves. Had they said that rel perm is a function of saturation, we are given these nice curves in textbooks. Would have been great if that was the case. But rel perm is not just a function of saturation, it's a function of capillary number, which is flow velocity, viscosity ratios, pressure gradient. Rel perm is a coefficient in a linear equation between flow rate and pressure gradient. And we find that it's a function of pressure gradient. So it's not a linear equation. So what kind of equation is it? Relationship is it? And as I said, it's supposed to be between 0 and 1. But there have been measurements of rel perm done by a group from Shell in the sandstone samples, rel perm up to 14. How is that possible? What is causing this? How can we explain that? So in terms of the initial premise, it doesn't work. It also doesn't model phenomena that we observe. This is a very well-known phenomena, infiltration of water into dry soil. If you take, these are experiments done in Heidelberg by Hassanizadeh. If you take a homogeneous sand, this is a sandbox, and then you introduce water on top. Now, you would expect that the water will go in as a front, diffuse front. That's what also the equation tells you. The equation I showed you, they are diffusion type equation. Will give you a diffuse front, but we invariably get these fingers develop, so it's unstable, and the water flows along these fingers. Moreover, you would expect that the saturation is higher up there and becomes very low at the tip of the finger. It is not. The equation, the saturation looks like this. If you plot that, it is indeed some high, and then at the tip of the finger, it goes up, becomes very large at the tip of the finger. It's non-monotonic. The equations I showed you will never become unstable and will never give you non-monotonic behavior. Others have measured this saturation. This is by DiCarlo for different flow rates, and then they see that indeed at the tip of the finger, the saturation goes up. We have done recently ourselves measurements in a dual energy gamma system that we have, and we take this is similar to DiCarlo's experiment. We take a glass tube, fill it up with sand, and we did experiments with dry sand, 4% and 6%, and then we rain water on top of it, and with this system we can measure saturation at various points along this, and we also have transducers, we can measure pressures along the tube. And here are the saturation variation with time at a position 20 centimeters below the top for different flow rates, and you can see in all cases the saturation is 0. The finger arrives, it's a large saturation at the tip of the finger, and then goes to the saturation behind the finger. And if you look at the pressure of the wetting phase, it's also non-monotonic, large at the tip of the finger. So how can we have a theory that can explain this and provide non-monotonic behavior for the two-phase flow? So that's what I'm going to introduce, but first I'll talk about microscale capillarity theory. Most of us are familiar with that. And then what should be the macroscopic capillarity theory, provide the ways of explaining the equilibrium curves I showed you, and also the non-equilibrium curves, and then what the theory should be for the non-equilibrium conditions, and I will get to the really, truly extended Darcy's law at the end. The capillarity theory at the microscale. This is the equation we all know. It's called Young-Laplace equation. And students ask me, what is now really capillary pressure? Is it Pn minus Pw, or is it 2 sigma times kappa m? Kappa is the curvature. So if you have a circular tube, you can write this equation as 2 sigma over the radius of tube times cosine of theta, theta being contact angle. So you know this equation. So is it this one or is it this one? And I say these are actually 2 equations given here. The second part, this is the true definition of capillary pressure as a property of a meniscus with a given surface tension and curvature. The first part of this equation is actually a balance of forces. And this balance of forces, in general, doesn't it won't be just this. This is what you get only under a static condition, if the meniscus and the liquids don't move. Otherwise, we will have viscous forces that are present within each liquid. They are also present at the meniscus. And the balance of forces for a meniscus at the point on the meniscus is actually this form, where this is the viscous forces from the two liquids on the two sides of the meniscus. And these are non-zero when there is flow. You know that. There is, for the viscous liquids, we have Newtonian viscosity, and that would be the forces that are shown here. And so how important this term is, let me show you the results of a work by Sheng and Zhu, published in the literature. They look at they solve the equations for the movement of two liquids in a tube. So what is shown here is half of a tube. This is the center of the tube. This is the wall of the tube. We have a non-wetting phase and we have a wetting phase. And there is a meniscus between them. Now, obviously, if there is no flow, again, we have this equation. But when there is flow, we know that there is a no-slip boundary condition at the wall for viscous liquids. So in fact, the velocity distribution, as you may know, looks like this, that it is 0 at the wall, and then it's parabolic, maximum in the middle. That's what we know for a tube for each phase. It's the same on the other side. But the interface velocity at the wall is not 0. The interface slides through the tube. So there is a slip boundary condition there. So they find this out, and they show that in that region, there are actually circulation of liquids taking place, and that circulation will be accompanied with dissipation, which means that the same viscous forces that I showed you are active here. And I can show you this video from Maria Dragila. Let me see, I hope it works. There you go. Okay, so this is a wetting phase displacing the non-wetting phase. Do I say that correctly? No, the non-wetting phase displacing the wetting phase. And these are colloids in there that are showing their movement, shows the movement of the fluid. And let's look at it again. See, there is those circulations taking place here. And that will give you dissipation. And that will give you, therefore, this extra term. This is what Sheng and Zhou have obtained, that the pressure difference is equal to the capillary pressure. I have to go back. Is equal to capillary pressure. This is this term here. But then some extra term due to flow. Q is flow velocity, and mu is the viscosity. A and B are constant. And the plus and minus are for when there is drainage and when there is imbibition. So even for a single meniscus, under flow conditions, we don't necessarily have Pn minus Pw equal to Pc. But of course, in a porous medium, the flow is very small, so this term is most probably, in most cases, negligible for a single meniscus. But not when we look at a very large number of collection of pores. So what would be the capillary pressure at the macroscale? Average pressures, differences between liquids. Once again, under quasi-static conditions, capillary pressure is everywhere constant. Fluid pressures are everywhere constant. So if you average constant values, you get those constant values back. So this can be derived under static conditions, but it cannot be otherwise derived under dynamic conditions. And so also the question is, even if we derive and say that capillary pressure is simply difference between pressures of the phases, is it just a function of saturation or not? Now, in order to study this, we have done a systematic upscaling from microscale to larger scale to derive equations instead of just extending it in an empirical way and throwing terms in there. How have we done that? We have used what we call a rational thermodynamic approach or averaging thermodynamic approach. At the microscale, when you have two-phase flow, you have three phases. So alpha phase could be solid, and beta and gamma could be two fluids. What we also have, we have interfaces between these three phases. They are shown here. And if you have a point within a bulk phase or on the interface, there are different balance laws valid for those points. And what we do, we write the balance laws for each balance of mass, momentum, and energy for points within each phase, but also for points on the interface. And then we average these balance equations using averaging methods and obtain macroscale conservation equations. And then we provide constitutive equations at the macroscale. And in doing so, we use the second law of thermodynamics. I'm not going to get into that, but this will provide us with governing equations of momentum. So that would be, as you know, Darcy's law is a reduced form of momentum equation, and we derive them for two-phase flow without making, say, heuristic assumptions. Here are the macroscale conservation of mass for each phase. We get this equation: porosity, saturation, and mass density, and flow rate, and exchange of mass between phases. Typically, we assume that density is a constant in compressible phases. Then we get this volume balance equation, and we are neglecting the mass exchange between the phases, which I gave you in the beginning of the talk. That's what we use. It is fine. But we also get a balance of macroscale balance of mass for interfaces. And that looks like this, where gamma is the mass present within an interface per area of the interface, and then average over an REV. And A alpha beta is the area of interface per unit volume of REV. So A alpha beta, you can write, is the total area of interface alpha beta divided by volume of the porous medium. We call this the specific interfacial area. What does it represent? I'll show that in a minute. So if we assume that the mass density of an interface is also constant, if we do not work with surfactants, we can make that assumption, then we get an equation for the evolution of the interfaces, which is very similar to that, but now for the interfaces. And the interfaces also have an average velocity. When we do drainage, for example, we see that the interfaces are moving. I'm going to show that to you through the porous medium, and you can define an average interface for them. How about capillary pressure? Now, everything is really energetically driven. If you take a sugar cube and you bring it in contact, a dry sugar cube, bring it into contact with tea, you see that tea rushes into that sugar tube, fills it up, expels the air. Why? It's because the sugar-air surface energy is much larger than water-sugar surface energy. So the system will go to a lower energy state if its saturation goes up. And that's exactly the definition of capillary pressure that comes out of our theory, because this H is the Helmholtz free energy. So we have a definition for capillary pressure, but of course, we don't work with Helmholtz free energy. But what we do realize comes out of the theory that this Helmholtz free energy is a function of saturation, but also interfacial area and mass density of fluid and temperature. So this suggests that the capillary pressure is a function of not only saturation, which we always assume, but also interfacial area. Or you can inverse that and say interfacial area is a function of saturation and capillary pressure. And then the next question would be, what does the theory say about PC and difference between pressures? Are they equal or not? So I'll get to both of them. But what does this interfacial area dependence mean? Let's go back to these capillary pressure curves. As I said, we have these kinds of scanning curves. We actually have data points. These are all equilibrium data points. In fact, we can have more equilibrium data points. You can actually fill up that whole domain with equilibrium data points. This is telling you that the capillary pressure is not just a function of saturation. You are missing another state variable, something that is also changing even for a given saturation. And that thing is actually the configuration of the liquids for the same saturation. You can have many different configurations when you do drainage and imbibition. And I'm going to show you from studies that we have. But what this therefore is saying, that instead of a curve, we really have a surface. If we plot interfacial area, saturation, and capillary pressure data points, then we get a surface shown in black here. And if you project that surface onto saturation capillary pressure plane, which is what we usually do, then you get this whole domain, the red domain, which are the points on that surface. So the theory is saying that we should work with a capillary pressure saturation interfacial area surface, that when you measure these three parameters, they fall on a surface, and they fall on a single surface for drainage, imbibition, scanning, whatever. Is that the case? Well, I'll show you that in a minute. But first, I wanted to show you this figure from lattice-Boltzmann simulations, where it shows distribution of water between glass beads at the same saturation. So these are the same amount of water. And look at how different the configuration is during drainage and during imbibition. Now, obviously, you cannot say, and the capillary pressures are very different, so you cannot say, if I know saturation, I will know capillary pressure. What you also need is this interfacial area that's now very different between the two. So is that really the case? Do we have a single interface? Now, I show you first the studies from what we call computational models. This is a pore network model where we represent the porous medium in the computer conceptually by an assembly of pores in all three different directions, big pores being pore bodies, the smaller pores being pore throats, and then we can always model flow of liquids and fluids in these network of pores. We can do static experiments like I showed you in a pressure plate apparatus to measure capillary pressure. And we can do that by saying, okay, this is saturated with water, and we put the non-wetting phase here, increase its pressure. Will it go into these pores or not? We have Young Laplace equation to check that. So we can determine the saturation and the distribution of phases in this set of pores. We typically work with 50, 60,000 pores. And then we can determine not only saturation, but also we can see what the areas are. We can calculate the areas because we know where the interfaces are in different pores. So we can calculate PC as a function of saturation for main drainage, the scanning curves, and so forth. This is imbibition, the scanning curves. We can then also calculate interfacial area as a function of saturation, and that is also hysteretic. And then we put all these together, we get a surface, three-dimensional surface. These are based on imbibition points, and we can get the drainage points, and this gives you a surface. When we compare them to each other, the difference is less than 10%. So we can say for all practical purposes, we have one single interface for all those data points under drainage and imbibition. We can fit a formula for an interfacial area as a function of saturation and PC, simple quadratic by quadratic formula, and to describe that surface. Others have done this also numerically. Lattice-Boltzmann studies from Porter et al., and all the data points fall on a single surface. These are also a list of other studies where they have done not only computational modeling, but they have done column experiments, and they have shown that they get indeed a almost single surface for drainage and imbibition and all the scanning curves. They are also done a lot of micromodel studies. I'm going to show you our own micromodel study that we have been doing. A micromodel is a two-dimensional network of pores that we create either within a glass plate, but here we have created this two-dimensional network of pores within a PDMS material, which is a transparent polymeric material. These are network of pores, which are, and the liquids we used were water and fluorinet. This is a non-aqueous liquid. And the overall dimensions were 35 millimeter long, 5 millimeter the size. These are input reservoir and output reservoirs, so we can inject liquids through this. And then the nice thing is that we can put this under microscope and see where the liquids are and where the interfaces are and how they move with time. The number of pores here, we had different sizes of micromodels, 2,000 to 6,000 pore bodies, and 6,000 to 24,000 pore throats. And the pore size, the mean pore size from 40 to 70 micron, and the depth was of the same size as the mean pore size. Here is one of our micromodels. That is the pore network. These are the input and output reservoirs. We put this not under microscope because with the microscope you can see only one square part of this. We want to see the whole thing as a function of time. So we have a setup where we can do that. We put the micromodel here between these two reservoirs. Then light goes through it, by a prism becomes horizontal. There is here a lens for light enlargement if we need, and then goes into this box. And what we have in that box is a set of beam splitters. What a beam splitter does, it makes from one image two perpendicular images. And here we have 3 beam splitters, and we get 4 perpendicular images of the original image with a slightly less intensity. And then we have 4 cameras. These are number 7 shown here. There is one on the back. And each camera zooms on 1/4 of our micromodel so that we can see the whole micromodel as the processes occur dynamically. Here is a video of a drainage and imbibition process. Let's see it works. Okay, so the dark is the non-wetting phase. As you go, you see it enters, displaces the wetting phase, and then we do imbibition. We put in back the wetting phase, and then we repeat. So we go, and we can do this statically, and we can do it dynamically. And each time, we know at any given time exactly where the interfaces are. So with image analysis, we can calculate interfacial area, we can calculate saturation, and we know the pressures at the boundaries. So if we look at, I'm going to show you 2 images for the same saturation, 24%, but then one would be for drainage from the same area, and one for imbibition. Look how different these fluids configurations are. We need to somehow parameterize this difference in fluid configuration with the same saturation, but under different conditions. And we believe interfacial area does exactly that. It's a macroscale measure that is a lump measure of how our interfaces are distributed. These are data points for the static capillary pressure saturation curves. As you can see, lots of data points during imbibition and drainage and the scanning curves. And then, as I said, we also measure interfacial area, so we can plot the surface of area, saturation, capillary pressure. We can do that for drainage, and then we can do that for imbibition, and we have compared them, and the difference is less than 10%. And we can also fit a surface, an equation to the surface, a unique surface. This is the average between the two. So it does tell us that the macroscale capillary pressure is related not only to saturation, but also interfacial area. And that actually describes hysteretic effects. And in fact, there are other properties that are hysteretic. Here I talked to 2 colleagues here. One of them is looking at thermal conductivity of unsaturated soil. And that is also hysteretic. And the other colleague is looking at suction stresses when looking at deformation and poromechanics. That is also hysteretic. And that is also because of the way the 2 liquids are distributed. And I believe interfacial area will also help in those cases. So how about dynamic conditions, non-equilibrium conditions? What is Pn minus Pw equal to? And what we find is that this is a linearized equation, that Pn minus Pw is equal to capillary pressure, but under dynamic conditions, when saturation changes, there is an extra term. This is a non-equilibrium term. And the faster the drainage will be, the more important this term will be. And there are many industrial porous media that we are dealing with large and fast drainage processes. So this tau is a coefficient, and we have measured this, done experiments to measure it. Here is a setup of a column experiment where we have, again, hydrophilic and hydrophobic transducers at these three elevations. We have also TDRs to measure saturation. I'm not going to go through the detail of this experiment, but here we could do a static experiment, equilibrium experiment, and we can do dynamic experiment, where we inject the non-wetting phase at a large pressure. Here are the results. The lower curve is the curve we get. Wait, I need to get my pen back. So the lower curve is the equilibrium curve. So under static or equilibrium conditions. The other curves are we get at larger and larger injection pressures. And you see that they are significantly different. They even become non-monotonic in form. And what is interesting and important is that we can use these curves to determine that coefficient tau. Because you see, this is the equation. Pn minus Pw is any of these curves. But they are happening at larger and larger saturation rates. Pc is this red curve. The difference between them is tau ds dt. So this is tau ds dt. We measure saturation as a function of time, so we can also determine ds dt, so we can calculate tau. Tau values have been calculated for that particular experiment is a weak function of saturation. Others have also calculated this, and this additional term will allow you to have non-monotonic behavior. In fact, here is a plot of Pn minus Pw at one of those elevations, the column, as a function of time. The blue dots are the data point. You see, it's non-monotonic, and if you use the standard theory where tau is 0, we get a diffusive type curve. It's not non-monotonic. And if you assign values to tau and do simulations, we get that non-monotonic behavior. So it does model, also these equations are unstable and will give you fingers in the infiltration into dry soil. So in the conclusion on capillarity theory is that the capillary pressure is not just a function of saturation, but we have a saturation interfacial area capillary pressure surface. And this, once you determine it under static conditions, you can always use it for dynamic conditions. And then what you should do for dynamic is not to say Pn minus Pw is equal to Pc, but we have that tau ds dt, the dynamic term. The interfacial area is important not only for describing flow, but also, actually, it's more important if you have kinetic mass transfer. Nowadays, we are very much interested in CO2 sequestration. And one of the mechanisms for having the CO2 kept down there is for the CO2 to dissolve in brine. The dissolution occurs through this kind of interfaces. It doesn't matter how much brine is back there, it's mattering how much contact area is between them, the amount of interfacial area. And in fact, therefore, for kinetic mass transfer or kinetic heat transfer, you really have to include interfaces in order to be able to properly model that. Now we can ask, how about Darcy's law? How should that be extended? What is the extended Darcy's equation? And this is what the result we get. Where this G is not pressure, that is Gibbs free energy. And Gibbs free energy, again, is a function of mass density, interfacial area, saturation, and temperature. We know that everything occurs because of energetic gradients. You have mass transfer not because there is a gradient in concentration, but there is a gradient in the chemical potential. But chemical potential depends on concentration. So we write Fick's law in terms of the gradient of concentration. But the actually driving force is the gradient of chemical potential, which gives free energy. And it's the same here. Now, if you have a saturated porous medium, that means one single fluid filling all the pores, then there are no interfaces in there. So that is 0. Saturation will be 1. And then the gradient of Gibbs free energy will be related to the gradient of pressure. We can prove that. We can show that. Which means that for saturated porous media, this rho del G becomes del P. We get Darcy's law. But if it is unsaturated, then the gradient of Gibbs free energy will have these other terms. We have extra terms in here. And this actually can explain why rel perm is bigger than 1 and why rel perm is a function of pressure gradient. Because if you neglect these terms, then what should take their role? That would be rel perm, because density is constant, viscosity is constant, saturated permeability is constant, so their role has to be taken up by rel perm. And therefore, rel perm will be a function of pressure gradient or flow velocity. And moreover, if these terms have a positive contribution to flow, that means rel perm must be bigger than 1. But if we include those terms, then rel perm will hopefully behave as a function of saturation, will not depend on pressure gradient and flow rate. We have studied these extra terms. I'm not going to go into that. We can get an equation also for movement of interfaces, also depending on the gradient of Gibbs free energy of interfaces. So we have an equation similar to Darcy's law for interfaces. And when we put all the equations together, we have the following set. We have equations of volume balance for each phase. You know that already. This is the extended Darcy's law. We have an equation for the evolution of interfaces. We have also an equation for the velocity of interfaces, the equation for non-equilibrium capillarity theory, and that the capillary pressure, saturation, and area are related to each other. The red terms are new terms in here. And you may say, wow, this is hopeless. We cannot solve all of this. Maybe you're right. I'm not saying that's what we should do. I'm saying that we must have a complex theory for a complex system, and then we go back and say, okay, in our case, this term and that term is not important, it's negligible. Fine, we neglect it. But we have to have the term first before we can neglect it. And that's how it should go. And in fact, I would believe that for primary drainage, indeed, we don't need these extra curves. And the standard theory will terms, and the standard theory will probably work well. But otherwise, we may need those terms. We have done pore network modeling to evaluate these extra terms in here, and these coefficients, what they are. And with our micromodel experiments, we are hoping that we can also determine them for a, say, physical porous medium. With that, I would like to end, and I'll be happy to answer any questions you may have. Thank you. Thank you. Yes, please.
Philosophical question. How do you go from the micro model with a phenomenon like capillary fingering to a continuum model, even with your extra interfacial area?
First of all, in the micro models, we haven't looked at fingering yet. We will look at situation that, as you saw, that we have a distributed movement of the two phases. But if you do,
Stochastic medium, how do you think you would capture the kinds of phenomena like fingering?
okay, then the fingering will be modeled, the kind of fingering you mean in that soil, dry soil. That is modeled by this dynamic capillarity term. Mathematicians have proven that Richards equation is unconditionally stable. But when we add to the Richards equation this extra dynamic term, the tau dsdt term, the equations become conditionally unstable. Under certain conditions, they will indeed give you fingers developing. Yeah, what's the question back there? Yes.
Air water or interfacial area remains a really difficult property to measure or even to determine computationally. And so in processes where the theory is improved by including that term, how do we deal with that limitation? How do we get that number?
Yeah. Well, that's a valid question, but that's a question that we have now because few people have thought of measuring interfacial area. In the beginning of 20th century, when Darcy Buckingham came, Buckingham came with his extension of Darcy-Buckingham equation, and he said that we should measure water content. The same question was asked: how do you measure water content in the field? And because at that time, the only way water content could be measured was volumetrically. Now we have 10 different ways of measuring water content using electrical conductivity, TDRs, neutron scattering, tomography, many ways to measure water content. I am sure that if we can see that this interfacial area plays a role as a state variable to model two-phase flow and the configuration of liquids, people will come up with ways of measuring it. There are already coming up with that. Interfacial tracers have been introduced to measure within a soil sample the amount of interfaces, and they are looking into the role of electromagnetic signals that are hysteretic. So they are most probably, for the same saturation, you get different signals going through. So they are probably related to the configuration of. So there are ways that I'm sure will come up. And even it is perhaps possible from the capillary pressure saturation data points that we have, with all hysteretic, with all the scanning points, they include actually information about interfaces. Maybe we can do inverse modeling using those terms. I don't know. I'm hoping that more clever people will come up with ways of determining, if I convince them, this is an important state variable.
A couple questions. The tau in that tau in the SDT, is that the same Newton law of viscosity tau?
No, it's not.
So how do you determine that?
Let's see if I can quickly go back to one of the last here. So that's what I explained here, that we can measure a static PCS curve. That's this term. We also, in our experiments, measure the two pressures separately. That's these curves. The difference is tau dSdt. S we know as a function of time, because we measured that with TDR. So we know the dSdt, so the only unknown is tau. And we have determined that.
What does that vary with?
It does vary with saturation. There are people are still studying this, what the form of that saturation is. That's why when we did a modeling, we use different forms of dependence of tau and saturation to do the modeling, but all of them give you this non-monotonic behavior. Other questions? Yeah.
So if I were to think of interfacial area as a state variable, would that be under like laboratory theoretical conditions? Because if you were to add vegetation or microbes into the equation, the structure of soil would continue to change. So wouldn't the interfacial area continue to change under those circumstances?
Yes, it does. It is a state variable for a specific soil. As soon as because it is very much related to the distribution of the two liquids at a given saturation. And that distribution will change if you change the pore space geometry or add other things, roots in there. So it will be like any other state variable, depends on a specific material. Yes, sir.
If you can go back 2 slides to the power. Yes, right here. And under this slide, what kind of a pressure did you use to induce this dynamic condition?
These are the the injection pressure in this system. We inject the non-wetting phase under drainage. We also did experiment for imbibition. These are drainage. So this is 20 centimeter. That is equivalent to 2 kilopascal water pressure in here. Now the entry pressure was about 6,000 Pascal. So with 8,000 Pascal, we start to go in. And we put in pressures of 20 kilopascal, that's 20,000, 30,000, and so forth.
Right. So my question to you is, at what kilopascal you don't see any difference? Is that if you start from 10?
That is, if we change 20 kilopascal, as you can see here, will basically displace the phase completely. Only if we change pressure incrementally. So for example, if these data points here, these are incremental changes. So from here to here, we really did this. Now, if you say, I want to go from this point to this point, then you will be doing this. And then the relationship between Pn minus Pw and Pc will be this curve here. So it's about Pn minus Pw, how far is it from Pc? And that indeed is determined by this kind of curve I'm showing you. So the closer they are to each other, the smaller that difference will be. Imagine you have a soil sample, given saturation, given Pc, equilibrium. Now you change Pn. Now Pn minus Pw is gone up. Pc hasn't changed yet because saturation has to change. And the drainage takes time. And that's exactly this kind of behavior. You increase Pn minus Pw, and then saturation has to decrease, and then we reach another equilibrium. In the meantime, Pn minus Pw and Pc are not equal, and they are separated by tau ds dt.
Right. So the bigger the step you change, more tau you will
No, the tau will be the same. The bigger ds dt becomes. Because if you put a large pressure, injection pressure, you're going to change saturation with a large value within a short time. That's what exactly it does. You can see that here. We increase that. Now saturation has to go from here to here. So that is the saturation time change within a given time. That makes this ds dt large. Tau is a property of the system. What changes each time you put a large injection pressure? It's like draining soil. You can drain it very slowly, or you can drain it fast. That will change your ds dt.
So if you go to the next slide, so what's the relationship if you plot them as a curve? Do you see some pattern
Between
SW and tau?
tau is a function of saturation. So I've given, so these are the kind of effect you see. As the saturation decreases, here tau has decreased, but yeah, it's not really non-monotonic. These are within the range of experimental error we have. You cannot say this is a significant difference. You can see that it's 10 to the 5, and then here, yeah, 1,000 change over 1,500 change, 15,000 change.
If I use 20 kilopascal versus 50 kilopascal.
Tau will not change. When you have different injection pressures, you only change the SDT. And the corresponding, so you can go back to this equation. You have a bigger injection pressure, this becomes bigger. This stays the same, this stays the same, this becomes bigger.
So for fat finger versus a thin finger, can this predict the phenomenon? If you use a lot of pressure, suddenly increase the interfacial pressure between the fine sand and coarse sand, you create different finger sizes?
I believe so. These are, maybe I have here results from a simulation where it shows here. These are simulations using this tau ds dt term, and this is infiltration of water into dry sand. And you see these fingers develop. Saturation is high at the tip of the finger. So this is Richard's equation. The only addition is tau ds dt term is added there. And how thick or thin these fingers are, I don't know. Other questions?
Okay, well, thank our speaker
Oh.
again.
All right.
One more, if it's okay.
Oh, one more.
The characteristic curves that we were just looking at, the different injection pressures, it was pretty clear that the injection pressures seem to affect the curves mostly near higher saturations. You know, the difference between the static curve and the dynamic curves were different, more different at higher saturations. Yet the interfacial area is probably more important at lower saturations, around 20–30%. So is there how do we explain that difference, or do we need to?
Oh, this is not related to interfacial area. These effects are related to that dynamic effect, that the Pn minus Pw and Pc are not the same when there are water movement or phase movement taking place. It's not related to interfacial area. So if you recall these set of curves that I showed you in the beginning, here. Let me give them all. Okay. So this hysteretic behavior, that is explained by interfacial area being an additional state variable that explains that at a given saturation you can have different capillary pressure. These curves, they are a consequence of a fast process, a fast drainage process. And therefore, the tau dSdt term takes care of, defines these extra terms. These are transient curves. They are time dependent. And that difference between these curves and the corresponding static curve is by tau dSdt. And during drainage, dSdt is negative, so minus tau dSdt is positive. That's why these dynamic curves are larger, higher than the static curve. During imbibition, dSdt is positive, minus tau dSdt is negative, so dynamic curves fall below the static curves. And that's exactly what we also see in experiments. Okay. All right, thank you very much for your attention and for questions.